Welcome to the deep dive. Today we're really getting into CRISPR technology. It's quite a story. Started as just this weird thing scientists saw in bacteria, and now, well, it's revolutionizing medicine. We've got a great report here mapping out that whole journey. So our mission really is to walk you through the key moments, the big scientific jumps, and where things stand now with CRISPR as a therapy. We want to cut through some of the noise. This field moves so fast. We'll give you a clear, engaging look at what matters most.
Okay, let's kick things off. The very first sort of whisper of CRISPR was way back in 1987. What was that first finding?
Yeah, it's fascinating because it wasn't like they were looking for it. Yoshizumi Ishino and his team, they were studying phosphate metabolism in E. coli, standard stuff, and they just happened to notice these unusual repeating bits of DNA near a gene called IAP. What caught their eye was the pattern, these identical DNA segments repeated, but with unique sequences separating them. And those separators were all the same length. Very structured, but totally mysterious. Nobody knew what they did, and they didn't look like anything else known at the time. It's odd. A little flag in the data saying, look here later.
Pretty much, a biological question mark. It hinted something interesting was going on, but yeah, it took years to figure out.
Okay, so fast forward a few years, 1993. Francisco Mojica enters the picture. What did he find?
Mojica was looking at a different microbe, an archaea called Halophorax mediterranei.
Right.
And he found similar repeats there. It was really the first deep dive into these specific structures. Initially, there was this idea from his supervisor's work, actually, that maybe they helped organize DNA during cell division.
Makes sense as a starting hypothesis.
It did, but it turned out to be wrong for these repeats. Still, that wrong turn was important. It pushed Mojica to keep investigating what they were doing.
And finding them in archaea, too, not just bacteria, that must have suggested they were more fundamental.
Exactly. This wasn't just an E. coli quirk. It looked like a basic feature in the prokaryotic world.
And then came the genomics boom. Suddenly we're sequencing everything. Did these repeats start showing up more often?
Oh, absolutely. Late 90s, early 2000s, genome data exploded, and bam, these sequences started appearing in lots of different bacteria and archaea genomes. Mojica saw the pattern, the connection between all these findings. He initially called them SRSRs, short, regularly spaced repeats.
SRSRs, yeah, it doesn't quite have the same ring to it, does it?
Not really. So Mojica teamed up with Ruud Janssen to standardize things. In 2001, they proposed the acronym CRISPR, clustered, regularly interspaced, short palindromic repeats. Much catchier.
Definitely.
And Janssen was the first to actually use CRISPR in a paper in 2002. And that same year, Janssen's group made another huge discovery. They found the genes that always seem to hang out near the CRISPR arrays.
The Cas genes.
That's them. They identified the first four, Cas1 to Cas4. And critically, they noticed these genes were only in prokaryotes, bacteria and archaea, not eukaryotes like us or in viruses.
Okay, so that strongly hinted at a function specific to those simpler organisms.
Exactly. Something they did that we didn't.
Right, so we've got the CRISPR repeats, we've got the Cas genes nearby. What was the next piece of the puzzle? Mojica had another key insight, didn't he?
He did, and this was really the breakthrough moment for understanding the function. Mojica looked closely at the spacer sequences, those unique bits between the identical repeats, and he realized something amazing. Their DNA sequences matched sequences found in bacteriophages.
Viruses that infect bacteria.
Exactly. And also archaeal viruses and plasmids, those little mobile DNA elements. So the spacers weren't random junk. They were like genetic mugshots of past invaders.
Wow. So that led to the big hypothesis.
Precisely. Mojica proposed CRISPR is an adaptive immune system for prokaryotes. The idea was a bacterium gets infected, survives, and it grabs a piece of the attacker's DNA. It then weaves that DNA fragment, the spacer, into its own CRISPR array, like adding a photo to a most wanted gallery.
A genetic memory.
Exactly. A memory that lets it recognize and fight off that same invader if it shows up again. He proposed this in 2003, but it met some resistance initially. Took until 2005 to get it published in the Journal of Molecular Evolution.
Seems obvious in hindsight, but I guess it was a radical idea back then.
It challenged existing ideas, yeah. But then came the experimental proof. And interestingly, it came from the food industry.
The dairy industry, right. Yogurt bacteria.
That's the one.
Yeah.
Philippe Horvath and his team at the food company Danisco, this was in 2007, they were working with Streptococcus thermophilus, a bacterium crucial for making yogurt and cheese. They wanted to know why some strains resisted phage attacks that wiped out others. And they showed experimentally that resistant bacteria had spacer sequences matching the phages they were resistant to.
Uh-huh.
And even better, they showed that when bacteria survived a new phage attack, They actually integrated new spacers from that specific phage into their CRISPR arrays. They could even deliberately add or remove spacers and change the bacteria's phage resistance.
Direct proof. That's fantastic. And Rodolphe Barrangou was key there, too.
Yes, he was a major player in that work. And you can see the commercial value immediately protecting valuable bacterial cultures for DuPont, who acquired Danisco later, nature's own antivirus software.
Okay, so the immune system idea is confirmed. Now, how does it actually work? How does the bacterium fight back using this memory? That brings us back to the Cas genes.
Right. Remember the Cas genes Janssen found? Well, in 2005, Alexander Bulletin's group identified a really important one in Streptococcus thermophilus, a big gene coding for a protein they predicted could cut DNA, a nuclease.
Cas9.
Cas9, the molecular scissors. And Bulletin also spotted something else crucial nearby, the PAM sequence.
Protospacer adjacent motif.
Yeah, that short DNA sequence right next to the target site in the invader's DNA. Cas9 needs to recognize that PAM sequence to know where to cut. It's like a landing signal.
Okay, so Cas9 is the cutter, PAM is the landing signal. How does it find the right place to land and cut?
That's where the CRISPR array itself comes back in. In 2008, John van der Oost's lab figured this out. They showed that the CRISPR array, with all its spacers, gets transcribed into RNA molecules, little copies. Specifically, each spacer becomes a small RNA molecule called a CRISPR RNA, or cRNA.
And these cRNAs are the guides.
Exactly. Each cRNA contains the sequence matching a specific past invader. It acts like a guide, leading the Cas proteins, including Cas9, to the matching DNA sequence in any new invader.
So CRISPR array stores the memory, cRNAs act as guides, Cas9 does the cutting, but only if the PAM site is nearby.
You got it. A really elegant defense mechanism.
Incredible. Okay, so the bacterial immune system is basically figured out. Then comes 2012. This seems to be the year everything changed.
2012 was absolutely pivotal. This is when the focus shifts from understanding the natural system to harnessing it. The landmark paper came from Emmanuel Charpentier and Jennifer Doudna. Working with the CRISPR system from Streptococcus pyogenes, they showed in vitro, in a test tube, that they could take the key components and use them to cut specific DNA targets. They figured out the role of another crucial RNA molecule, trucRNA, which pairs up with the cRNA.
The dual RNA guide complex.
Right. This dual RNA complex works with Cas9 to find and cut the DNA. But the real breakthrough, the absolute game changer, was realizing the system was programmable. Meaning you could give it any DNA target sequence you wanted.
Yeah.
Just by changing the sequence in the guide RNA.
Wow. So you could direct the Cas9 scissors to cut essentially anywhere in a genome.
Precisely. And they showed you could even make multiple cuts at different locations. It wasn't just understanding anymore. It was engineering, reprogramming nature's tool.
That's huge. And we should also mention Virginijus Jushainis. His team was right there too, weren't they?
Absolutely. It's important to recognize his contribution. Kanis and his group in Lithuania independently showed the same thing, programmable DNA cutting with Cas9. They actually submitted their paper to Cell Report slightly earlier in April 2012, but it faced some delays and ended up being published in PNAS that September, shortly after the Doudna-Charpentier paper in Science.
So, parallel discovery. It underlines how ripe the field was for this leap.
Definitely. And Kanis was later rightly recognized for his work, sharing the Kavli Prize.
Okay, so programmable DNA cutting is demonstrated in a test tube. The next obvious step. Can we make it work inside living cells, especially complex eukaryotic cells like ours?
That was the billion-dollar question, and the answer came incredibly quickly. In January 2013, just months after the in vitro work, 2 labs independently reported success. Feng Zhang at the Broad Institute and George Church at Harvard. They both showed that CRISPR-Cas9 worked for genome editing in mouse and human cells.
That fast. Incredible.
It really kicked the doors wide open. This demonstrated CRISPR wasn't just a bacterial tool. It could potentially edit the genes of any organism. The CRISPR revolution had truly begun.
And Zhang's lab sharing the tools widely really fueled that fire.
Massively. Making the plasmids and protocols available through Agene allowed labs everywhere to jump in and start experimenting. It democratized the technology very quickly.
And one more refinement made it even easier, right? The single guide RNA.
Yeah, that was another smart simplification. Remember the cRNA and trachRNA pairing up? Researchers figured out you could fuse those 2 RNA molecules together into one longer single guide RNA, or sgRNA. It still did the same job, guiding Cas9 to the target, but it was just simpler to design and use one molecule instead of 2. Made the whole system much more user-friendly.
Okay, so the toolkit is refined. It works in human cells. The therapeutic potential is suddenly very real. When did we see the first steps towards clinical trials?
Things moved incredibly fast on the clinical front, too. The first trials began around 2016, mostly in China initially. They were focused on cancer immunotherapy, taking a patient's T cells, editing them with CRISPR to make them better cancer fighters, and putting them back, often targeting lung cancer.
And in the U.S.?
The first U.S. trial started in 2019 at the University of Pennsylvania. Similar idea, editing T cells, but for patients with myeloma and sarcoma. And crucially, around 2018, the first company-sponsored trial kicked off for a genetic disease. That was CTX001 for sickle cell disease.
Using CRISPR to fix an inherited condition.
Exactly. That marked a really significant step towards treating the root cause of genetic disorders.
Which leads us right to Casgevy, the first approved CRISPR therapy. A massive moment.
Absolutely huge. Casgevy, the scientific name is ixagamglogin autotemcel, or AGACL for short, got approved for sickle cell disease and beta-thalassemia. First in the UK in late 2023, then the US, Europe, Canada followed.
How does it work exactly?
It's an ex vivo therapy, meaning outside the body. They take hematopoietic stem cells, the cells in your bone marrow that make all your blood cells out of the patient. Then, in the lab, they use CRISPR-Cas9 to edit a specific gene in those stem cells. They target an enhancer region for a gene called BCL-Lib A.
And disrupting that gene does what?
BCL-Lib normally acts like a switch, turning off the production of fetal hemoglobin after birth and turning on adult hemoglobin. In sickle cell and beta-thalassemia, the adult hemoglobin is faulty. So by using CRISPR to break that BCL-Lib switch, the edited cells start producing fetal hemoglobin again.
Which is functional and can compensate for the faulty adult version?
Exactly. Then they infuse these edited stem cells back into the patient where they hopefully engraft in the bone marrow and start producing healthy red blood cells with fetal hemoglobin.
A potential cure, essentially.
For many patients, it's looking like a functional cure, yes. It's a powerful validation of the whole approach. And Lyfgenia, another gene therapy, got approved for sickle cell too, showing real momentum.
And the pipeline isn't just for blood disorders. CRISPR's being aimed at many other single-gene diseases.
Oh yes, lots of activity. There are trials and research targeting the CFTR gene for cystic fibrosis. For Duchenne muscular dystrophy, people are trying different CRISPR strategies, like skipping over the faulty parts of the gene or directly correcting it. Labor congenital amaurosis, a form of inherited blindness that saw the very first use of CRISPR in vivo.
Meaning the editing happened directly inside the patient's body.
Correct. In that case, injecting the CRISPR components directly into the eye to target the CEP290 gene. We're also seeing work in hemophilia, targeting clotting factor genes. And there was a really remarkable case treating a baby with a rare metabolic disorder, CPS1 deficiency, using a personalized CRISPR approach. Shows the potential reach.
It's incredible. And back to cancer, CAR T cells are getting a CRISPR boost too.
Definitely. CRISPR is making CAR T cell therapy, which already had great successes, even better and potentially more widely applicable. Researchers are using CRISPR to, say, knock out genes in the T cells that normally put the brakes on their activity, making them more persistent cancer killers.
Taking the brakes off the immune response?
Kinda, yeah. Or using CRISPR to insert the CAR gene, the bit that recognizes the cancer cell, into a very specific, safe place in the T cell's genome. This might lead to more reliable and controlled CAR T function.
More precise engineering.
Exactly. And another big goal is using CRISPR to create allogeneic, or off-the-shelf, CAR T cells.
Meaning not needing to use the patient's own cells each time?
Right. Editing donor T cells so they don't cause graft versus host disease or get rejected by the patient's immune system. That could make CAR T therapy much more accessible and faster to deploy. Lots of trials are testing these ideas now.
Okay, beyond genetic disorders and cancer, infectious diseases, how can CRISPR help there?
This is a really exciting area too. One approach is to directly target the virus's genetic material. Research is ongoing for viruses like HIV, trying to use CRISPR to cut the viral DNA out of infected human cells, or to disable critical viral genes in things like SARS-CoV-2.
Wow, directly attacking the virus's code.
Yeah. Another angle is tackling antibiotic resistance, using CRISPR, maybe delivered by phages, to specifically target and cut the genes that make bacteria resistant to antibiotics, or even just kill the resistant bacteria themselves.
A targeted weapon against superbugs.
Potentially. And CRISPR is also being developed for diagnostics, rapid sensitive tests to detect specific viruses or bacteria. There was even a first trial using CRISPR-enhanced phage therapy for urinary tract infections caused by E. coli. Early days, but promising.
The applications just seem to keep branching out. Cardiovascular disease, autoimmune.
Yeah, frontiers are expanding. For cardiovascular disease, there's interest in using CRISPR in vivo to edit genes in the liver to permanently lower things like bad cholesterol or other risk factors like LPA, by targeting genes like ANGPTL3. For autoimmune diseases, maybe editing immune cells to make them less reactive. Regenerative medicine, enhancing tissue repair. And there's a whole other layer with epigenetic editing using modified CRISPR systems not to cut DNA, but to switch genes on or off, controlling their expression without changing the underlying sequence. So controlling gene activity rather than rewriting the code itself. Exactly. Lots of potential there for fine-tuning cell function.
The pace is just stunning. But let's be realistic, it can't all be smooth sailing. What are the big hurdles still? Delivery seems like a major one, getting CRISPR into the right cells in vivo.
Delivery is absolutely a key challenge. You have these amazing molecular scissors, but how do you get them precisely where they need to go in the body and only there? People are using modified viruses like AAVs or lentiviruses as delivery vehicles. They're pretty good at getting into cells.
But viruses come with their own potential issues.
They do. Immune responses, potential for integrating into the genome in unwanted places, limits on the size of the CRISPR system they can carry. So there's huge interest in non-viral methods too. Lipid nanoparticles, or LNPs. Those are the same fatty bubbles used for the mRNA COVID vaccines. LNPs can package the CRISPR components, maybe as mRNA or protein, and they've shown real promise for delivery, especially to the liver. Other methods include electroporation, using electrical pulses to open temporary holes in cells, or even direct microinjection for certain applications. Each has pros and cons.
And once delivered, our immune system might still react to the CRISPR components themselves, particularly Cas9.
Yes, immunogenicity is a big concern. Cas9 is a bacterial protein, after all. Many of us might actually have pre-existing antibodies against common Cas9s, like the one from strep pyogenes, just from past exposure to those bacteria.
So our bodies might attack the therapy.
They might. It could reduce effectiveness or cause side effects. And repeated doses could certainly trigger a stronger immune response. So researchers are working on strategies, using Cas enzymes from different bacteria that humans haven't encountered as much, developing off switches or ways to degrade Cas9 quickly after it does its job, or even cloaking the Cas9 protein somehow.
Okay, delivery and immunity. What about accuracy? Making sure CRISPR cuts only where intended. Off-target effects.
Precision is paramount. You absolutely want to avoid unintended cuts elsewhere in the genome. A lot of progress has been made here. Scientists have engineered high-fidelity Cas9 variants, versions of the protein that are much, much less likely to cut at sites that only partially match the guide RNA.
Making the scissors pickier.
Exactly. There's also exploration of entirely different CRISPR systems, like Cas12a or Cas13, which have different properties, different PAM requirements, maybe different off-target profiles, and techniques like paired nicking. Instead of making one double-strand cut, which can be messy to repair, you use two guide RNAs and two Cas9s engineered to only nick one strand each at nearby locations.
Two single cuts instead of one double cut.
Right. The cell repairs these nicks much more cleanly, and it requires both nicks to happen close together, greatly increasing specificity.
And even with all these precautions, we need to think long-term, right? These are permanent changes to the genome.
Absolutely crucial. We need long-term follow-up. For Casgevy, for instance, patients will be followed for 15 years. We need to see if the therapeutic effect lasts. Does the fetal hemoglobin production stay high? And we need to watch carefully for any delayed side effects. The biggest theoretical concern, though considered very low risk with current methods, is causing unintended genetic changes that could, down the line, contribute to cancer.
Oncogenesis.
All right. We also need to understand the impact of mosaicism. What happens if not all the target cells get edited or get edited correctly? How does that affect long-term function and safety? These are critical questions as the therapies mature.
And the CRISPR toolkit itself keeps evolving beyond just cutting. You mentioned base editing and prime editing.
Yes, the field isn't standing still. The original CRISPR-Cas9 creates a double-strand break and relies on the cell's repair pathways, which can sometimes introduce small errors, insertions, or deletions. Base editors are different. They use a deactivated Cas9 one that can still find the target DNA using a guide RNA, but can't cut both strands fused to an enzyme that can directly change one DNA base letter into another, like changing a C to a T or an A to a G.
More like a pencil and eraser than scissors.
That's a great analogy. It allows for very precise single-letter corrections without that double-strand break, potentially safer and more predictable for certain types of mutations. And then prime editing is even more versatile. It also uses a nicking Cas9 fused to a different enzyme, reverse transcriptase. It can make small insertions, deletions, and all 12 possible base-to-base conversions. It essentially guides the synthesis of a corrected DNA sequence directly into the target site.
So even more sophisticated gene surgery.
Exactly. These newer tools offer potentially higher precision and fewer byproducts for certain types of edits. And scientists are constantly discovering new natural CRISPR systems too, like Cas13 that targets RNA instead of DNA, or Cas3 that shreds DNA over longer distances. The toolbox is getting incredibly rich.
Which brings us to the ethical and societal side. This power raises huge questions.
Immense questions. Accessibility and cost are major ones right now. These first therapies are incredibly expensive. How do we ensure equitable access?
Yeah, Casgevy's price tag is over $2 million.
Exactly. Then there's the ongoing debate about germline editing, making changes to sperm, eggs, or embryos that would be passed down through generations. Most countries currently prohibit this for reproductive purposes due to safety and profound ethical concerns.
The designer baby fear?
That's part of it, yes. But also just the unknowns of altering the human gene pool permanently. Regulatory agencies like the FDA, EMA, Health Canada play a critical role in carefully evaluating safety and efficacy before approving any therapy. And public discussion, engaging with the societal implications, is absolutely vital to ensure this powerful technology is used responsibly.
So wrapping up, it's just been this breathtaking journey for CRISPR, from, as we said, a bacterial puzzle
to a technology that's already delivering approved medicines for devastating genetic diseases. It's truly astounding.
The collaboration, the key insights from so many researchers along the way, Ishino, Mojica, Janssen, Horvath, Barrangou, Bulletin, van der Oost, Doudna, Charpentier, Zhang, Church. It's a testament to scientific discovery.
Absolutely. Building block by building block.
And while challenges remain delivery, safety, cost, ethics, the sheer pace of innovation suggests we're just scratching the surface of what CRISPR might achieve.
The potential feels enormous.
Which leaves us with a final thought for you, our listeners. Having this kind of precise control over the very code of life, what does that mean for us? What future possibilities, what responsibilities does this technology place upon humanity? Definitely something to ponder.